In our last lecture, we have discussed about some of the terms that are frequently used in the biological manuscripts or manuscripts that are related to biological. Let us use those terminology to understand some of the one of the concept of one of the concept to analyze the data, that is analyze the data, and that concept is GSEA, that is gene set enrichment analysis. Yesterday we have understand what is meant by wild type and knockouts. So when we have suppose we have the proteomics data of wild type and knockout, and we need to compare what is different in knockout cells compared to the wild type. In order to use this GSEA, what first it will does is suppose we have the list. List of gene that is expressed list of gene that is expressed in wild type and list of gene that is expressed in knockout. And we also have the differential expression data, some quantitative analysis, such as if someone has used mass spectrometry, then area under the curve of that gene, some or any aspect that determine that can quantitate the amount of that gene present in that sample. And when we have these two different samples, what we do in GSEA first is to prepare a ranked gene list. Ranked gene list. And how this list is prepared? List is prepared by comparing the same gene of different sample and see whether that gene is overexpressed or underexpressed or equally expressed in the knockout. And then list is prepared like this. Like here we will have upregulated upregulated genes, and at the down we have down-regulated genes. Down-regulated genes, and the sequence, and this as we go from up to down, the upregulation decreases, and in the middle we have something where it is not upregulated or down-regulated, and below this we have the genes that are down-regulated in the knockout. And after that, when we have the ranked gene list, let's consider this as a chart, and we need to determine whether the specific pathway is upregulated or downregulated. What we do is we check where we check the genes involved in that specific pathway are present in this list or not. If they are present in this list, we will mark them. Suppose one pathway, this is gene ontological pathway, and we need to check for protein folding. It is not necessary that we need to have some pathway that is already defined. We can use a custom pathway based on the experiment, or instead of using pathway we can use something different, like like sets of kinases reactions, sets of kinases, and sets of genes that are present in some organs to check whether that genes are upregulated or downregulated in knockout or not. It depends on the interest. It depends on the interest of the experiment. Okay, so let us consider this example. We are using GO is called gene ontological. It is generally used term in the biofield, and any gene ontological for protein folding, it has the genes that are involved in the protein folding. And if we check where these genes are present in the list, and if it seems like this, then we can judge from the first impression that it seems like it is upregulated. It is upregulated in the knockout compared to the wild type. And if we check for some another pathway, GO ubiquitation. Suppose this is just an example I am taking, and if it shows here, mainly here, 1-2 could be here, but majority of the protein should be if it is shown in bottom side of the list, then we can assume that it is downregulated. So this and if some pathways are equally distributed among the gene list, then it is neither upregulated, downregulated. So this is just the first impression. After that, we need to do some quantitative analysis. We need to do some multiple testing in order to statistically prove that our pathway is significant, in order to statistically provide evidence that whether our pathway is upregulated, significantly upregulated or downregulated in knockout or not. And about that statistics, we will discuss this. We will discuss about that in our next video. So that's all for today. Thank you.